Today I'm going to be doing something a little different. I will be rereading Brain and Behavior, an Introduction to Behavioral Neuroscience by Bob Garrett and Gerald Hough or something like that. This is the second time taking this class, and so I've read everything already, but there was a chapter that we skipped over that first time around, so I'm going to record it. And so we are going to be on Chapter 13, Intelligence and Cognitive Functioning. Some people are calling Cambridge's theoretical physicist Stephen Hawking the most brilliant person living today. Following in Einstein's footsteps, he developed theories of the origin of the universe that are altering the way scientists think. He lectures around the world, mixing high-powered physics with a keen sense of humor. He has achieved all of this despite Lou Gehrig's disease, the amyotrophic lateral sclerosis, or ALS. A degenerative disease that impairs voluntary movement. Confined to a wheelchair and able to make only small facial movements, he writes and speaks by moving a cursor on the screen of a computer equipped with a voice synthesizer. When we consider people like Hawking, we are forced to wonder what makes a person more intelligent than another. Is it the genes, the upbringing, hard work, or opportunity? Is it the ultra-intelligent brain in some way? Unfortunately, we have a problem with that presumption, with the presumption of who is smarter. Is Hawking more intelligent than Einstein? Or has he just had the advantage of more predecessors' accomplishments in which to build on? To Marilyn von Savant, is Marilyn Voss Savant smarter than her husband because at the age of 10 she made the highest score recorded on the intelligence test, while he had trouble testing well enough to get into medical school? Or is he smarter because he invented the Jarvik artificial heart? We cannot attempt to understand the biological basis of intelligence without having some appreciation of our limitations in measuring it or even defining what it is. The nature of intelligence. There are many ideas about what intelligence is, which is the first clue that we don't have consensus about what it is. Most definitions say something to the effect that intelligence is the ability to reason and to understand and profit from experience. What we think intelligence is, that is what we think intelligence is. The problem comes when we try to translate the abstract definition into behaviors that would indicate a presence of intelligence. That is what we must do to measure intelligence first, which is the first step towards determining its biological basis. And over here we have a picture of Stephen Hawking. His great intellect resides in a body that can communicate only by moving a cursor on a computer screen. Okay, so what does intelligence mean? Understanding how we measure intelligence is important because we are in effect defining intelligence on what tests measures as because we are in effect defining intelligence as what that test measures. Okay. The measure of intelligence is typically expressed as the intelligence quotient, or IQ. The term originated with scoring on early intelligence tests designed to use for children. The test produced a score in the form of a mental age, which was divided by the child's chronological age and then multiplied by 100. The tests were designed to produce a score of 100 for a child performing at the average for his or her chronological age. The scoring is completely different now, partly because the tests are more extended to adults who do not increase consistently in intellectual performance from year to year. The base score is still 100, and a value selected arbitrarily and preserved artificially by occasional adjustments to compensate for any drift in performance in the population. Most people are near the average. Figure 13.2 shows, with relatively few people at either of the extremes. For example, only 2% of the population score above 130 or below 70. The first intelligence test was devised by Alfred Binet in 1905 to identify French school children who were in need of special education, or need of special instruction. Predicting school performance is still what most intelligence tests do. The intelligence tests have found their greatest use in a school setting. The correlation between IQ and school grades typically falls in the range of 0.40 to 0.60. If you have forgotten what the correlation coefficient means, please revisit the pages 112 and 113. However, IQ is also related to job performance, income, and socioeconomic level, and negatively to juvenile delinquency. Critics believe that the scores of traditional intelligence tests are closely related to academic performance or to higher socioeconomic levels, mostly because the tests were designed to reflect that kind of success. According to these critics, the test overemphasizes verbal ability, education, and Western culture. A few tests are designed to be culture-free, like the Raven Progressive Matrices. Matrices, yeah, okay. The tests are mostly nonverbal. The tests require no experience with particular culture, and they have obvious advantage for testing people from a variety of different cultural and language backgrounds or with impaired understanding of language. Some people also believe that these tests give them a better representation of pure intelligence. Claiming that true intelligence is more than what the tests measure, these critics often point to instances where practical intelligence, or street smarts, is greater than conventional intelligence. For example, a young British street vendor conducted their business as they were adept from performing calculations. They were unable to perform in a classroom setting. In another study, experts racetrack gamblers use highly complex algorithms involving 7 variables to predict racetrack odds, but their performance was unrelated to their IQ. In fact, 4 of them had IQs in the low to mid 80s. More recently, Robert Sternberg compared the scores of presidential candidates like George W. Bush, Al Gore, and Bill Bradley, and made on the verbal section of the Scholastic app and made on the what they made on the SAT. Okay, that's what we're saying. Okay, when they applied for college, the SAT had many similar items of those on conventional intelligence tests. 2 of the candidates scored above average, and the college applicants, but not markedly so, and no one had a score that was below average. To Sternberg, their success raises a question about the narrowness of intelligence tests. Sternberg in 1988 argues that intelligence does not exist in the sense where we usually conceive of it, but it's a cultural invention to account for the fact that some people are able to succeed in their environment better than others. Perhaps intelligence, like the mind, just a convenient abstraction of we invented to describe a group of processes. If so, we should not expect to find intelligence residing in a single brain location or even neatly defined network of brain structures. And to the extent in which we find processes or structures that they are directly involved in intelligence, their performance may not be correlated with scores on traditional intelligence tests. All right, the figure 13.2 shows the distribution of IQ scores in the population. Gifted is and average is, standard distribution looking thing. Okay. The structure of intelligence. Another controversy is the critical to biological understanding of intelligence, is whether intelligence is a single capacity or a collection of several independent studies or abilities. Intelligence theorists tend to fall into one of 2 groups, lumpers and splitters. Lumpers claim that intelligence is a single unitary capability, which is usually called the general factor, or simply G. General factor theorists admit that there were separate abilities that vary somewhat in their strength of the individual, but they place much greater weight on the underlying G factor. They point out that the person who is high in one cognitive skill is usually high in others. So they believe that the measure of G is adequate by itself to describe a person's intellectual ability. General intelligence is sometimes assessed by several overall IQ scores from traditional intelligence tests, such as the Wechsler Adult Intelligence Scale. Those 11 subtests measure more specific abilities. But many G theorists prefer to use other tests, like the Raven Progressive metrics, because they emphasize reasoning and problem solving and are relatively freer of influence from specific abilities such as verbal skills. Splitters, by contrast, hold that intelligence is made up of several mental abilities that are more or less independent of each other. Therefore, they are more interested in scores on the subtests of standard IQ tests and scores from tests on specific cognitive abilities. They may agree that there is a general factor, but they give more emphasis to separate abilities and differences among them in an individual. An accurate description of a person's intelligence would require the scores of all of the subtests of these abilities. These theorists point out to cases of brain damage in which one capability is impaired without affecting the others, and to a savant's exceptional ability in a single area. Splitters disagree with each other, though, on how many abilities there are. And a review of intelligence tests identified more than 70 different abilities that could be measured in available tests. Biological origins of intelligence. Okay, so with this background, we are now ready to explore the origins of intelligence. Based on our introduction, we will avoid two popular assumptions: that intelligence tests are the only way to define intelligence, and that intelligence is a single entity. Instead, we will consider performance and achievement in addition as additional indicators of intelligence, and we will examine the evidence of a biological basis for the general factor, and then consider the relationship between brain structures and individual abilities. The brain and intelligence. So how intelligent brains are different. Okay. Is a more intelligent brain different in any identifiable way from other brains? Anyone asked the questions that would naturally wonder how Albert Einstein's brain was different from other people's. Fortunately, the famous scientist's brain was preserved, and it has been made available from time to time to neuroscientists. In cursory examinations, it turned out to be remarkably unremarkable. In fact, at 1,230 grams, it was almost 200 grams lighter than the average weight of the control brains. The number of neurons did not differ from normal. Studies have disagreed about whether the neurons were more densely packed or the cortex was thinner. Perhaps because the samples were taken from different locations, one study found a higher ratio of glial cells to neurons in the left parietal lobe. The comparison brains averaged 12 years younger than Einstein's at the time of death, and we know that glial cells continue to proliferate throughout life, but the number of glial cells was not elevated in Einstein's right parietal lobe or in either frontal sample. Each of Einstein's hemispheres was a full centimeter wider than those of control brains due to the larger parietal lobes, and were intriguing variations in some of the parietal gyri. These abnormalities are interesting because the parietal lobes are involved in mathematical ability and visual spatial processing, and Einstein reported that he performed his mathematical thinking not in words but in images. Remember, though, that in Chapter 12 we saw that the London taxi drivers had enlarged posterior hippocampi, and no one suggested that the large hippocampi explained why they became taxi drivers. In fact, a later follow-up study found that drivers who qualified to end their training, who qualified at the end of their training increased in hippocampal volume, but drivers who failed to qualify did not. Whether Einstein's large parietal lobes or intense mathematical ability came first is uncertain, assuming that they are related. It is interesting that the strongest finding in the examination of Einstein's brain seems to be related to specific ability rather than overall intelligence. British researchers John Duncan and his colleagues sought the location of general intelligence in brains of more ordinary folk. They use these tests to require reasoning that are known to correlate with general intelligence more than with any specific ability. Although verbal and spatial tasks are different patterns of activation, prefrontal activation is common in both. In both tasks. Okay, so the authors conclude that general intelligence may be located there, but general intelligence is likely more complex than what happens in one brain area. A team of researchers scanned brains of 241 individuals with brain damage and correlated the location of their lesions with their scores on a set of intelligence subtests chosen to measure G. They found that G requires a distributed system, a network that spans from the parietal and temporal lobes, and they concluded that G involves brain's ability to pull together different types of processing, such as visual spatial processing and working memory. The ability to integrate these functions depends on the quality of connections between the areas, which is highly heritable, up to 84% in some parts of the brain. Brain size. Popular wisdom is that smart people have bigger brains, and that is true, to an extent, at least A meta-analysis of 88 studies, including more than 8,000 individuals, or brains, I guess you'd say, found a correlation between intelligence and brain size for about 6%, or 0.24 squared, of people's differences in intelligence. Lest we give too much importance to brain size, Einstein's 1,230 gram brain was a bit more than the 100 grams under the average for a male of his age. Nevertheless, brain size is a factor in intelligence, but we can't generalize this across species. Elephants have much larger brains, and not many people think elephants are as smart as they are. Most people, more important than brain size is our compact primate neural. The more important than brain size is our compact primate neural organization that packs more neurons into a smaller space. And on top of that, we have the largest brains of all the primates, and so far as we know, the largest number of neurons. So if our brain size accounts for just 6% of our differences in intelligence, then other factors must be more important. Men's brains are 11% larger on average than a woman's, yet men and women score equally on intelligence tests. Men have a size advantage, but women apparently compensate with different structural organization. They have a greater ratio of gray matter to white matter than men do, which increases their number of neurons. Women also have stronger connections between the hemispheres and more complex convolutions. I'm like highlighting everything, so bear with me. And they rely on different parts of the brain than men to achieve comparable results in the intelligence testing. Gray matter and white matter volumes are more specific measures of overall size of the brain and are both predictive of intelligence. The correlation of gray matter with IQ has been measured at 0.37. In men, the correlation is strongest in the frontal and parietal lobes, and in women, show the strongest gray matter relationship in the frontal area, including Broca's area. White matter volume, which indicates the extent of connectivity within the brain, has a correlation of 0.26. Variations of white matter volume are more important in women for women's cognitive performance than men's. Note, in figure 13.5, the widespread distribution of significant areas suggests that intellectual ability depends on a broad network of neural structures. So let's look at the figure. It says brain areas involved in general intelligence. A view of the cortical surface left and the subcortical areas right, shows a G-related network extending through much of the brain. Cool. I really do enjoy this book. Okay. Processing speed and efficiency. Cognitive processes require a person to apprehend, select, and attend to meaningful items from a welter of stimuli arriving at the sensory organs. When a person must retrieve information from memory, relate the new information to it, and then manipulate the mental representation of the combined information, all of this takes time. Okay. Back in 1883, when there were no intelligence tests, Sir Francis Galton attempted to relate measures of intelligence achievement, like course grades and occupational status, to people's reaction times. But he could not find the relationship. Later, researchers have shown that IQ scores are correlated with reaction time, and even more so with nerve conduction velocity. Neural conduction speed depends on the integrity of the white matter tracks. White matter integrity is correlated with intelligence and explains 10% of the variance. White matter integrity declines with age and intelligence along with it. Faster nerve conduction may make its contribution through improved processing efficiency. White matter integrity and nerve conduction velocity are vital in determining how well information travels throughout the brain. Most important contributors to both, therefore, to transmission efficiency is myelination. which increases conduction speed and protects against crosstalk between the neurons. Humans have a greater proportion of white matter or myelinated processes to the gray matter than other animals, and varieties among individuals with the degree of myelination. In addition to myelination, speed of information processing and intelligence all follow a curvilinear time path, increasing from childhood to maturity and then declining in old age. One indicator of the role of brain efficiency in intelligence is that individuals who have higher IQs use less brain energy. This was indicated by a lower rate of glucose metabolism during a challenging task. Taking the computer game by playing the game of Tetris. And as you see in figure 13.7, the individuals with mild intellectual disability with IQs between 50 and 70 require 20% more of the neural activity to perform an attention-demanding task than individuals with IQ of 115 or higher. Let me see. Okay, so we have a bunch of brains showing activity in red. Correlations between gray matter and white matter volumes in IQ. Notice that the gray matter volumes is correlated with IQ in more areas in men than in women, and white matter volume is correlated with IQ in more locations in women than in men. The lighter colors indicate a stronger relationship. Interesting. Okay. You might think the more intelligent brain would be the more active one during a task, but remember that we are talking about efficiency. Improved technology, particularly the availability of diffusion tensor imaging, has shifted the conversation towards brain networks and network efficiency. After reviewing 37 imaging studies related to intelligence, Rex Young and Richard Heyer, 2007, proposed a parieto-frontal integration theory, or the PFIT. According to their theory, information processing takes place in 4 stages, primarily in the parietal and frontal lobes. Number 1, the sensory information has been processed in secondary areas it is passed on to. And number 2, parietal areas, which abstract the information and integrate it. And then 3, these areas interact with frontal areas in problem solving and evaluation. And number 4, finally, the anterior cingulate cortex selects the response and inhibits alternating responses. Subsequent studies had lent support to the theory, identifying these areas as central to intelligence and the parietal cortex as a main hub. In addition, the brains of more intelligent individuals were more efficient, with more neurons, more numerous interconnections within clusters of neural activity, and shorter paths between the clusters. Okay, specific abilities and the brain. Brain size, speed, and efficiency can reasonably be viewed as contributors to general intelligence. Now we will consider the evidence for individual components of intelligence. The statistical method called factor analysis has been useful in identifying these components. The procedure involves giving a group of people several tests that measure cognitive abilities that might be related to intelligence. The tests may be intelligence tests, or they may measure a more limited ability, such as verbal skills or reaction times. When clusters are calculated among all combinations of the tests to locate clusters of ability, when correlations are calculated among all combinations of the tests to locate clusters of ability that are more closely related to each other than with others. Performance on practically all tests and cognitive ability is somewhat related, which is consistent for the hypothesis of the general factor. However, factor analysis has also identified clusters of more specific abilities. These abilities have emerged frequently as a major components of intelligence: linguistic, logical, mathematical, and spatial. Several authors have argued that each of the cognitive abilities depends on the complex network of modules in the brain that has evolved in ways to support that function. In other words, they believe that the brain is hardwired for functions like mathematics and language. We have already seen examples of modular functions in earlier chapters. For example, the language or linguistic module is made up of a network of structures located mainly in the left frontal and temporal lobes. Spatial ability depends mostly on the functions of the parietal lobes. And an MRI study at the University of Iowa revealed structural differences related to male spatial superiority on the mental rotations test, a standard measure of spatial ability. Women in the study had greater gray matter volume relative to white matter in the parietal lobes, but this was correlated with poorer spatial performance. Men, by contrast, had greater cortical surface area in the parietal lobe, I assume, because I need to turn the page. Yes, lobes. after a correction for brain size, and that was correlated with better performance. Previous work has indicated that the greater cortical surface area translates to a larger number of neurons. Mathematical ability in humans depend on the prefrontal cortex and the parietal cortex. That fact by itself makes no distinction between mathematical ability and general intelligence. Most likely, the distinction lies in specifics of the interconnections and how the brain can recruit activity in the structures. For example, people who perform better on tests of basic arithmetic had greater cross-connection activity between the two parietal lobes during problem solving. In addition, the areas are activated selectively according to the task and progressively as difficulty increase. The left prefrontal cortex is most active when an individual performs calculations for memory, such as 2 times 4 and 2 plus 4. This area is probably the storage site for arithmetic facts. Both parietal areas come into play during estimation of values. This makes sense because visual spatial representations of quantity, such as finger counting and the number line, are an almost universal stage of learning exact calculation. Calculation recruits both left and right prefrontal areas and both parietal cortices. Activation increase further in the parietal lobes with increasing difficulty, incorporating an additional step to the calculation. And in the prefrontal areas, as speed demands increase. I guess that's where that period. Okay. Studies of brain damage support these conclusions. Individuals with damage to the left frontal language area can arrange a number in rank order and establish results, but they cannot perform tasks. Precise calculations. Those with parietal damage are impaired in the opposite direction. Catherine Keller and Vinod Menon in 2009 also observed frontal and parietal activation as their subjects did multi-step mental arithmetic. They also looked at the occipital areas. I glitched right there. The occipital areas found activation there, demonstrating that people recruit additional resources as needed. 4 clusters in the right hemisphere are more activated in men than in women. But this did not translate to greater calculation accuracy. So although the brain differences sometimes result in performance differences, once again we see evidence that males and females using different brain strategies to achieve comparable results. Obviously, neither of these areas is dedicated exclusively to numeric functions. This is consistent with the suggestion in Chapter 9 that the language areas may simply use the processing strategies that make them particularly suited to the demands of the task, rather than being dedicated to language processing. However, Wynne, in 1998, argues that brain has a specialized mechanism for numbers by pointing to the evidence that even infants and lower primates have an inborn ability to estimate quantities. The infants he studied saw 2 objects placed one at a time behind a screen. They acted surprised, which means that they looked for a longer time. And when the screen was removed to reveal the 3 objects, there was only one object instead of the expected 2. Rhesus monkeys tested in the wild responded to the task in the same way. Monkeys can rank order groups of objects that differ in number by touching their images on a computer monitor in the correct order, and chimpanzees learn to do the same with numerals. The accompanying research spotlight offers some additional insight into animal intelligence. All right, let's go there. Research spotlight. These are usually really interesting. We aren't the only tool users. Oh, we know this. This is okay. A chimp dines on termites. This is a picture of a chimp, and then a picture of the brain scan, the area. Yeah. So a chimp dines on termites fished from their mound with a stick. Humans have been fascinated with how intelligent animals' behavior can seem. Termites build earthen mounds that are so well ventilated that they stay cool under the African sun. Dolphins cooperate with each other to herd schools of fish into their pod mates' waiting mouths. The kanzi communicates with symbols. Alex mastered simple concepts, see chapter 9, and monkeys and chimpanzees can count. Especially intriguing is the concept of the tool use. Monkeys have long been known to fish for termites by punching a hole in the nest and inserting a small green twig that the termites can climb up onto in defense of their nests, but only to be slurped up by the chimp. Congo chimps add a unique twist. They fray the ends of the twig, enabling them to pick up 20 times as many termites. New Caledonia and Hawaiian crows cut and shape twigs and use them to extract food from crevices, and they have been observed to spontaneously shape wires into hooks to retrieve food. But Santino, a chimp in a Swedish zoo, may demonstrate the greatest ingenuity. Santino occasionally threw stones at visitors, but zookeepers weren't concerned because there was so little ammunition on his island. Then stone throwing increased dramatically, so caretakers watched to figure out where the stones were coming from. Each morning, Santino retrieved stones from the moat and placed them strategically for launching at the visitors. He was also seen dislodging chunks of concrete from the wall. After first tapping where there were cracks and listening for the hollow sound that meant a piece was loose enough to be removed. So what do these research observations tell us, aside from the fact that that guy wanted to be furry and did not like people? Sorry. Anyways, first, animals are more intelligent than we have given them credit for. And second, some of our capabilities aren't unique after all. But we also have a chance to discover where we are truly different. For example, while watching someone retrieve an object with a hand activates mirror neurons, they are simply located in humans and in monkeys. However, when humans see the object being retrieved with a tool, an additional area is activated in the anterior parietal lobe. Yet no such area for tool use has ever showed up in monkeys. Even the ones that were trained for 3 to 6 weeks to use the same tool. Apparently, we have developed some additional specialized structures, either through evolution or individual experience. An impartial observer would have to say that there is evidence for characteristics that contribute to an overall intelligence and for somewhat independent capabilities as well. As either or stance is not justified now, it makes sense to continue the two-pronged approach to looking for biological bases for both general intellectual ability and separate capabilities. Heredity and environment. As we saw in Chapter 1, that intelligence is the most investigated of all genetically influenced behavior. Our understanding of the genetic underpinnings of intelligence hardly lives up to the amount of effort. However, this is because intelligence is so complex and poorly understood itself, and because many genes are involved. In addition, environment accounts for half of the differences among us in intelligence, yet the environmental influences have themselves been clearly identified. Heritability of intelligence. Figure 13.9 shows that IQ correlations among relatives averaged from many studies and several thousand people. You can see that IQ is more similar to people who were more closely related. Separating family members early in life does not eliminate the correlation. In fact, identical twins reared apart are still more similar IQ than fraternal twins reared together. Increasingly, the relative influence of heredity increases with age. 41% in childhood, 55% in adolescence, and 66% in adulthood. Intuition tells us that environment should progressively take over genetic effects as siblings go their separate ways, and their experiences diverge. Yet J.R. Gray and Thompson suggest that the genes can influence intelligence, also influence the individual's choices of environment and experience. Researchers have documented genetic influence for several of the functions that contribute to intelligence, including working memory, processing speed, and reaction time in making a choice. Most of the differences among individuals in the major structural contributors to intelligence are accounted for by genetics. Established heritabilities in one twin study was 90% for brain volume and 82% for gray matter and 88% for white matter. General intelligence has higher heritability than more specific abilities, such as verbal or spatial abilities, which is an additional argument for a stronger biological basis for G. Locating the specific genes is another matter. Two genes have received special interests underwent accelerated evolution following our separation from chimpanzees. The ASPM gene is a major determinant of brain size, and while the PACAP precursor gene plays a role in neurogenesis, neural signaling, and may contribute to the formation of human cognitive abilities. But the search for genes has been fraught with one replication fail after another. One reason that effect sizes for individual genes are small is on the order of around 1%, which means that unless samples are very large, the genes will be missed. However, small samples often yield false positives that don't hold up under further scrutiny. So recently, a large consortium of researchers has been combining genetic data with brain scanning to locate genes responsible for structural foundations of intelligence. One of these studies has identified a genetic variant associated with brain size, the CLL of RS10784502. Its processor scored 1.29 points higher on IQ tests, and having two of the CLLs doubled the gain. A second study identified 24 genetic variations on 6 genes, which were related to structural integrity. Related to the structural integrity of major brain pathways. Some of these were associated with intelligence, and individuals with multiple of these variants scored several points higher than others did on intelligence tests. Another group identified a gene linking cortical thickness to intelligence. Individuals with particular variant had a thinner cortex in the left hemisphere and performed less well on intelligence tests. This variant, however, accounted for only 0.5% of the variation in IQ. The genetic controversy. The conclusion that intelligence is highly heritable has been greeted with unquestioning acceptance. The heritability of intelligence is controversial on a variety of fronts. These controversies illustrate both a pervasive misunderstanding of genetic influence and difficulty resolving questions of heredity. Critics fear that inheritance of intelligence implies that intelligence is inborn and unchangeable. Nothing could be further from the truth, however, as Weinberg points out, genes do not fix behavior but set a range within which a person may vary. By the way of illustration, height is about 90% heritable, yet the average height has increased dramatically over the past few decades due to improved nutrition. Similarly, IQ scores increase at the rate of 5 to 25 points just over a generation. See the term the Flynn effect. So test constructors have been adjusting the test norms occasionally to maintain the average of 100 points. Although the environmental causes have not been identified, such rapid increases cannot be due to genetic change. However, we know that the world is much more stimulating, information-filled place, and education has changed dramatically. And as Flynn points out, in the 1900s, the median year of schooling was 6 years old, compared to with 13 years now, the schools taught by memorization and recitation. Some researchers argue that the correlation of IQ among relatives does not mean that intelligence is inherited. They suggest, for example, that identical twins similar in appearance and personality lead others to treat them similarly even when they are reared apart. This similar treatment results in an intellectual development, although physical features and behavior do affect how others react to a child. There is little evidence that these responses in turn influence intelligence. To test this possibility, researchers compared IQs of twins who had either correctly or incorrectly perceived by their parents as fraternal or identical. If similar environmental treatment accounts for IQ similarity, then the parents' perception of their twins' classification should be more important than the twins' actual genetic classification. Instead, the studies showed that the only true genetic relationship influenced IQ similarity in the twins, not the parents' perception. In another controversy, a debate has raged since the 1930s about whether IQ differences between ethnic groups are genetically based. The question is not whether ethnic groups differ in IQ, but how much difference are due to heredity and to environment. Arthur Jensen, 1969, has argued that environment and socioeconomic differences are inadequate to account for the observed IQ differences among groups. He and Philip Rushton cite studies indicating that IQ differences are consistent around the world, with East Asians averaging 106 points, whether in the United States or in Asia, whites about 100, and African Americans at 85, and sub-Saharan Africans around 70. In addition, they say that the IQ gaps correspond to differences in brain size. Their position has practical as well as theoretical implications. For extreme hereditarians believe that intervention efforts like Head Start not only do not work, but also cannot work. Other intelligence researchers accounted that Jensen and Rushton ignored or misinterpreted the most relevant data. For example, African Americans and with more African ancestry scored high on cognitive tests as those with mixed ancestry. Another study suggests that socioeconomic class is more important than ethnic origin. It found that IQ runs about 20 to 30 points lower in the low social classes than in the highest social classes. Higher intelligence test results are not always found in Asians, and their higher academic achievement is typically attributed to cultural and motivational differences. The task force applied by American Psychological Association to study the intelligence debate concluded that there is not much direct evidence regarding the genetic hypothesis of IQ differences between African Americans, whites, and what little there is does not support the hypothesis. Environmental effects. Most intelligence researchers agree that intelligence is a result of joint contributions of genes and environment. It has been said that intelligence is 100% hereditary and 100% environmental because both are necessary. However, it has been more difficult than expected to identify which environmental conditions influence intelligence other than those that cause brain damage. We will give environmental influences more attention here than usual because intelligence is a good area for illustrating the difficulties in sorting out heredity from environment. One problem is similar to the one we have encountered in identifying specific genes. The environmental influences are many and, for the most part, individually weak. Even a twin study that looked at major environmental events such as severe infant and childhood illness failed to find an effect. The second problem is that environmental influences are often hopelessly confounded with genetic effects. For example, family conditions such as socioeconomic level and parental education are moderately related to the offspring's intelligence. But these characteristics also reflect the parents' genetic makeup, which they pass on to their children. Best way to demonstrate environmental influences is by environmental intervention. Although the Head Start program has produced long-term benefits in mathematics, educational attainment, and career accomplishments, the average increase is 7.42 IQ points compared to the controls eventually disappears. Okay, so they just initially. The Abacadarian Project, which begins at birth, produces IQ gains that were as strong 10 years later than those in the Head Start program after 2 years. Apparently, which is interesting. Apparently, intervention must occur at an early age. And a new early Head Start program now takes children at birth through the age of 5. Adoption has a better chance of demonstrating any environmental influences on intelligence because it alters the child's entire environment. Adopted children's IQs are more highly correlated with intelligence of their biological parents than with the intelligence of their adopted parents. But this does not mean that children's IQs do not go up or down according to the adoptive environment. When African American children were adopted to impoverished homes into middle-class homes, by the age of 6 their IQs had increased from the 90-point range for African American children in that geographic area to 106. The beneficial effect persisted decades later. This means that is there no genetic effect at all? No. In fact, the correlation between the children's IQs and their biological parents' education levels, used in the absence of IQ scores, increased over a 10-year period follow-up, while correlations with their adoptive parents' education levels decreased. It may puzzle you on how the children's IQs could be correlated with their biological parents' intelligence if the children's IQs had moved into an adoptive parent's range. Although we usually think that correlation indicates similarity between scores, this is not necessarily the implication. Rather, it means that the scores have similar rank orders in their groups. In other words, the parent with the highest IQ has the child with the highest IQ. The parent with the second highest IQ has the child with the second highest IQ, and so on. Now move the children into a new household and raise each child's IQ by 10 points. The biological parent with the highest IQ still has the child with the highest IQ, and the parent with the second highest IQ, well, you get the point. The correlation tells us that children's IQs are still tied to their biological parents' intelligence, as by an elastic string that can stretch but nevertheless affects how much the IQ can change. That elastic string in this case is the influence of the genes. Though it has been difficult to identify specific environmental influences that increase intelligence, we have had some success in identifying influences that have a deleterious effect. For example, one example is parental exposure to pesticides. An agricultural company in California whose mothers had high levels of organophosphate pesticide residue in their blood during pregnancy averaged 7 IQ points lower compared to children with mothers with the lowest measure. The exposed children scored lower on tests of working memory, processing speed, this makes me sad, verbal comprehension, and perceptual reasoning. Similarly, children with the highest levels of parental exposure of chlorpyrifos scored 2.7 points lower in IQ and 5.3 points lower on tests of working memory. Until it was banned for indoor use in 2001, chlorpyrifos is one of the most widely used insecticides for household pest control. Okay, before we switch the page, let's read about this application here. Enhancing intelligence and cognitive performance. Most of us feel the need for a bit of an edge as we deal with the demanding world. Drugs have long been a go-to solution. 34% of college students, for example, report using stimulants occasionally prescribed for ADHD or you know what I mean. The benefits of these drugs are modest, as the drugs can impair some types of cognitive functioning, while they also enhancing others. Manipulating genes might have been an alternative, but remember that the known genes have a very small individual influence. Neuroscience has created mutant strains of mice that were smarter at one task or another, but these mice also had their problems. One strain was overly fearful, the other solved problems well, but struggled with the simpler, solved some problems well, but struggled with the simpler ones. Faced with these realities, researchers have sought a more benign strategies. They have reported long-term improvement in solving mathematical problems following electrical stimulation to the scalp over the prefrontal cortex, and demonstrating cognitive gains after subjects have played video games that require extensive use of working memory. Furthermore, working memory training increased functional connectivity in the frontal parietal network. Given these successes, some researchers have suggested that the better question is not whether the technique worked, but whether the training regimens produced the best real-life benefits. Recently, 2 strategies have advanced to clinical trials. In studies of older, age 50 or over, individuals, both extensive training on various cognitive tasks and consumption of cocoa flavonols, nutrients in foods like cocoa, tea, grapes, and apples, for 3 months showed beneficial effect on cognition. Another study reminds us that the importance of early stimulation. Psychologists visited the home of Jamaican children aged 9 to 24 months who were stunted in growth, the bottom 5 percentile for height. Researchers played with the children weekly, provided books and toys, and taught the mothers better ways to interact with their children. Compared to the non-stunted control group, those who received early stimulation had better grades, higher IQs, showed less depression, and got into fewer fights. Later in adulthood, they had a 25% higher earnings. The bottom line is that there's no magic pill to increase intelligence, and the most promising approaches appear to be very basic. Early stimulation, adequate diet, and practice, practice, practice. Although childhood illness in general are not predictors of later intelligence, infection does seem to be important. A study in North Manhattan, New York, a community designed to identify risk factors of stroke, found that people with multiple risk-associated pathogens also had lower cognitive ability. Researchers in the University of New Mexico concluded that the level of infectious diseases is the best predictor of national differences in intelligence. After controlling for confounding variables, including education, climate, and economy, the correlation between the national average IQ and infectious diseases ranged from negative 0.76 to negative 0.82, depending on the method of estimating intelligence. The investigators believe that fighting off infections during childhood robs the body of energy needed for normal brain development. Not only might disease prevalence partially account for racial differences in intelligence, but also researchers suggest that the reduction in the incidence of infectious diseases explain some of the IQ increases seen in the Flynn effect. We want to understand the antecedents of intelligence to enhance cognitive functioning and prevent the disorders that diminish it. We have already seen in Chapter 12 that this is a daunting challenge in the case of Alzheimer's disease, and the accompanying application reveals that enhancing performance in normal functioning individuals is not so easy either, which we just read that application. Okay, deficiencies and disorders of intelligence. Intelligence is as fragile as it is complex. According to the list of conditions that can impair intelligence is impressively or depressingly long. To give you a feel for the problems... Sorry about that, forgot my glasses. To give you a feel for the problems that can occur in this most reverend of our assets, we will add a few thoughts to what we have already covered in Chapter 12 on aging. Take a brief look at intellectual disability, and then spend more time on autism in the recognition of its half-century-long challenge to neuroscientists' investigative skills. And finish with attention deficit hyperactivity disorder. So, question is, how much capability is lost by the elderly? Effects of aging on intelligence. In Chapter 12, we discussed the most widely known cognitive disorder of aging, Alzheimer's disease. Here we will limit our attention to normal declines in cognitive abilities that are associated with aging. One source of normal decline is the reduced activity of numerous genes involved in long-term potentiation and memory shortage due to age-related damage. Genes involved in synaptic functioning and plasticity, including those responsible for glutamate and GABA receptors and for synaptic vesicle release and recycling, are particularly affected. All right. Although intelligence and cognitive abilities do typically decline with age, the amount of loss has been overestimated. One reason is that people are often tested on rather meaningless tasks, like memorizing lists of words. When the elderly are tested on meaningful material, the decline is moderate. Another reason for the overestimation of these early studies were cross-sectional. Comparing people at one age with different people at another age, you have already seen that more recent generations have an IQ test performance advantage over previous generations. When comparison is done longitudinally, by following the same people through the aging process, the amount of loss diminishes. Although the loss depends on the type of intelligence measured, crystallized intelligence, skills and overlearned knowledge remain stable or improved. Vocabulary, for example, improves through the 7th decade. Problem-solving, reasoning, and the ability to process and learn new information make up fluid intelligence, which declines beyond the 3rd decade. Apparently, performance speed is particularly vulnerable to aging. Its loss turns out to be important. Shea, in 1994, found that statistically removing the effects of speed from test scores significantly reduced elderly individuals' performance losses. We saw earlier that working memory is especially important in intellectual capability. In the study of people ranging from 18 to 82, showed the speed of processing accounted for all but 1% of age-related differences in working memory. So you know that the determinant of general intelligence is the ability to integrate activity between the brain areas. Even in healthy aging, there is a loss of coordination in the default mode network. Proportions of the frontal, parietal, and temporal lobes that are active when the brain is at rest or focused internally rather than in the outside world. Activity in the default mode network is thought to represent preparedness for action. To the extent this coordination diminishes, cognitive ability does as well. Imaging reveals that the loss is due to the decline in white matter connections among the areas. In the elderly, those with better whole brain network efficiency, clustering and short path length, had higher processing speed and superior executive function compared to including impulse control and decision making. The brain does have ways of compensating, which as the increased metabolism that people see in subjects with mild intellectual disability and the recruitment of frontal areas with people with dyslexia during reading. Studies of elderly individuals who are aging gracefully indicate that they are holding their own through additional neural effort. For example, a memory task activated only the right prefrontal cortex in many adults and in older adults to perform poorly, but older adults who performed as well as the younger adults used both prefrontal areas to perform the tasks. The increase in frontal activity is in proportion to decreases in the other areas, which supports the idea that the shift serves as a compensatory function. Compensation may not be limited to frontal activity. It also has been seen in the parahippocampal area during memory recognition tasks. This less than efficient adaptation might not be the only pathway to graceful aging. Elderly subjects initially coped with a task via compensation, but just after 5 hours of training on the task, they show brain patterns that look like the young subjects. Some of the loss in performance is due to non-physical causes and is reversible. For example, older people often lack the opportunity to use their skills. In one study, aged individuals regained part of their lost ability through practice skills, and many of them returned to their pre-decline levels. They still had some advantages over controls 7 years later. Elderly people improved in memory test scores when their self-esteem was bolstered by presenting them with words that depict old age in positive terms, such as wise, learned, and insightful. Loss in performance that has had a physical basis may be reduced, if not reversed. Diet appears to be one factor. For example, in a study of 6,000 people over the age of 65, cognitive decline was 13% less in those who ate 2 or more fish meals per week, compared to people who ate fish less than once a week. Evidence in this study and others suggests that the important function was the overall pattern of fat intake. Vitamin supplements might also help. In elderly individuals with mild cognitive impairment, a combination of B vitamins reduced the rate of brain activity atrophy 50% and slowed cognitive decline compared with subjects given a placebo. The B vitamins slow gray matter atrophy by reducing homocysteine, a toxic compound that is elevated in people with a diet high in animal proteins. Interestingly, the sex hormones provide some protection against the cognitive effects of aging. In menopausal women, estrogen replacement therapy reduces the decline of verbal and visual memory, as well as lowering the risk of Alzheimer's disease. The importance of estrogen is supported by the fluctuations in performance that occur during the menstrual cycle. First, remember from Chapter 7 that women tend to be superior to men in some types of verbal tasks, and that men typically outperform women on tasks requiring spatial ability. During the part of the month when estrogen is high, women perform better on verbal tasks. When during menstruation, estrogen drops, and so does the performance on verbal tasks. But spatial performance improves. So how does estrogen produce these effects? Well, we're not sure, but we do know that neurons throughout the brain have estrogen receptors. Estrogen levels during the menstrual cycle are correlated with cortical excitability, increased glucose metabolism, blood flow in the areas involved in cognitive tasks, and responsiveness to acetylcholine, which is important in memory and cognitive functioning. Now, finally, untreated menopausal women are more impaired than women receiving estrogen replacement on tests of working memory, response switching, and attention, which tells us that estrogen improves functioning in the prefrontal areas. So what about the male species? Men who maintain testosterone production past the age of 50 have better preserved visual and spatial memory, better preserved visual and verbal memory and visual spatial functioning. The effects of replacement therapy have been variable, owing apparently to the form of testosterone preparation used. However, numerous studies have shown improvement in spatial, verbal, and working memory. Interestingly, testosterone improves only spatial memory. Additional memory improvement requires that testosterone be delivered in the form of dihydrotestosterone, which can be converted to estrogen in the brain by the processing of aromatization. I think I'm saying that right. Cool. Okay, so when it comes to cognitive abilities, we could attempt to consider estrogen a wonder drug. So losses are smaller than believed, but they differ across people and across skills. In addition, practice esteem enhancement and active lifestyle may slow cognitive decline during aging. Obviously, we cannot stereotype the older person as the person with diminished activities. Intellectual disability. Intellectual impairment was previously referred to as retardation, but that term has taken such a negative meaning in popular usage that practitioners and authorities are shifting to the term intellectual disability. Intellectual disability, or ID, is limited in the intellectual function of reasoning, learning, problem solving, and adaptive behavior in its developmental origin. The criteria set by the Diagnostic and Statistical Manual of Mental Disorders, 5th edition, DSM-5, are an IQ at or below 70, deficits in adaptive functioning, such as communication, social preparation, independent living, and the onset during the developmental period. Looking back at figure 13.2, you can see that 2% of the population falls into this IQ range. The lower percentage and a lower percentage would meet both criteria. Table 13.1 describes the areas of impairment that contribute to diagnosis and functioning impairment. Not only is any definition arbitrary, but it's situational and cultural as well. A person considered to have intellectual disability in our society might fare reasonably well in a simpler environment. The situational nature is illustrated by the fact that many individuals shed the label as they move from childhood from the childhood of academic failure into adulthood and demonstrate their ability to live independent lives. So let's look at this table here. Table 13.1, areas of impairment leading to a diagnosis of intellectual disability. Domain, conceptual, includes skills in language, reading, math, writing, reasoning, knowledge, and memory. Domain, social, empathy, social judgment, interpersonal communication skills, ability to make and retain friendships. Domain, practical, includes self-management in areas such as personal care, job responsibilities, money management, recreation, and organizing school and work tasks. Intellectual disability is an estimated prevalence of 1% worldwide, with a higher incidence in low and middle income countries. It can result from childhood infections, birth complications, extreme malnutrition, maternal alcohol abuse during pregnancy. Although intelligence is highly heritable, no reliable estimates of heritability of ID exist. Determining which genes are at fault has also been difficult, in part because ID is made up of a large number of disorders. Another problem is that while milder forms of ID involve common forms of inheritance, severe ID often results in de novo mutations. Those that appear spontaneously in an individual rather than being passed down from a parent. Nevertheless, about 400 genes have been convincingly linked to ID. However, this fact means that we can expect additional genes to have small effects and to be difficult to isolate. The most common genetic cause of intellectual disability is Down syndrome, with a prevalence of 1 in 700 births, usually caused by the presence of an extra 21st chromosome. Down syndrome typically results in individuals with IQs in the 40 to 55 range, although some areas are less impaired. Recall that the amyloid precursor protein gene that is involved in early onset Alzheimer's is located on chromosome 21, and that it was discovered because Down syndrome individuals also develop the amyloid plaques. 95% of people with Down syndrome have an extra chromosome, a complete trisomy 21, called complete trisomy 21. In 1% of the cases of the body's cells have an extra chromosome, but not all of them do. That is a mosaic trisomy 21. In a few cases, only an end portion is present and is attached to another chromosome. This is called translocation trisomy 21. A stain of mouse TS65DN engineered with a third copy of 55% of chromosome 21 has been a useful model in studying Down syndrome. As in people with that disorder, its glial cells secrete less of two proteins that support neuronal survival. Treating pregnant TS65DN females with these proteins eliminates the developmental delays that would ordinarily be seen in their offspring. Convincingly, the strategy could be used with pregnant women when amniocentesis, genetic testing of the amniotic fluid, reveals that the fetus has a third 21st chromosome. Another therapeutic possibility is postnatal correction of inadequate neuroepinephrine release in the hippocampus, a deficiency that is believed to contribute to learning impairment. After treatment with a drug that increased neuroepinephrine levels, TS65DN mice performed as well as normal mice. However, it is an oversimplification to focus only on the effects of chromosome 21 genes. DNA myelination differs by more than 10% between subjects with Down syndrome and controls, and the degree of myelination difference is correlated with cognitive functioning. Although they differentially methylated genes were found on other chromosomes, the authors attributed the hypermethylation to the presence of the third chromosome 21. Okay, and so we have figure 13.3, which says Down chromosomes and born this way. Chromosomes of a person with Down syndrome, female. The arrow points to the 321st chromosomes. Some individuals with Down syndrome are more fortunate than others. The documentary series, Born This Way, follows 7 young people with Down syndrome in Southern California as they pursue their various careers and dreams. It's showing all the different groupings of chromosomes, which I could never explain. Okay, moving on. Another frequent genetic intellectual disability is Fragile X syndrome, which is due to mutation of the Fragile X mental retardation gene, one gene, the FMR1. The FMR1 gene normally contains between 6 and 45 repetitions of the nucleotide sequence CGG. If the number of CGG repeats reaches 200, the gene is turned off and no protein is made. So Fragile X syndrome is the result. With IQs typically below 75, an intermittent number of CGG repeats results in a reduced protein production, accompanied by a slight increase in the chance of intellectual disability. Males are affected more often. When Fragile X does occur in females, the symptoms are usually milder. The FMR1 knockout mice usually numbers the unusual numbers of dendrites and immature spines indicate a failure to prune excess synapses. I'm going to try to say this one. Phenylketonuria is due to an inherited ability to metabolize the amino acid phenylalanine. The excess phenylalanine interferes with the myelination during development. So newborn infants are routinely tested for phenylalanine in the urine or blood, and intellectual disability can be prevented by avoiding foods containing phenylalanine. The artificial sweetener aspartame is a familiar example of a substance that is high in phenylalanine. Without dietary treatment, the individual is severely and profoundly disabled, with an adult IQ of around 20 points. The hydrocephalus, make sure I'm saying this right. Hydrocephalus occurs when cerebral spinal fluid builds up in the cerebral ventricles. The increased fluid volume crowds out the neural tissue, causing intellectual disability. As we see in Chapter 3, hydrocephalus can also be treated and caught early by installing a shunt that prevents the accumulation of the excess cerebral spinal fluid. Also, in that chapter, you learn that some individuals seem not to be harmed by the dramatic loss of cortex. In fact, half of those ventricles fill 95% of the cranium, have IQs over 100. The Zika virus. Zika virus, one of the infectious causes of intellectual disability, can have devastating effect on the brain development and intelligence. The accompanying in the news feature describes the effect of Zika on neural development. Let's look at it. In the news, Zika virus impacts neural development. During the summer of 2016, an outbreak of the Zika virus became an increasing public health concern when babies born with Zika-infected mothers showed smaller than normal brains, a condition called microcephaly. Even with the knowledge from dozens of early investigations, there are many unanswered questions regarding Zika effects. Although Zika infection has been clearly linked to microcephaly, studies still must be done to determine the mechanisms by which Zika impacts fetal development. In addition, not all infants are born to infected mothers show signs of microcephaly. Fortunately, the infant brain retains much of the fetal brain's plasticity, which leaves open the possibility that ways could be found to altering postnatal brain development, particularly in cortical areas, to compensate for the disruption caused by the virus. Tang and colleagues in 2006 have investigated how Zika affects the brain by infecting stem cells, then coaxing them to develop into neural tissue. The results were clear. Infection resulted in reduced overall neural cell numbers, both by increasing cell death and by disrupting the cell cycle. Other scientists have evaluated brain development using MRIs to see how often the fetal brain is impacted by the mother's infection. In a preliminary report on a sample of 48 fetuses, only 3 showed abnormal development. Other studies are planned to determine the long-term consequences of exposure and whether exposed babies born with normal-appearing brains might still be affected neurologically. Okay. Autism spectrum disorder. Autism spectrum disorder, or ASD, is a set of neurodevelopmental disorders characterized by social deficits, communication difficulties, and repetitive behaviors. In the DSM-5, autism spectrum disorder includes previously used diagnosis of autism, Asperger's syndrome, pervasive development disorder, and not otherwise specified childhood disintegrative disorder by the American Psychiatric Association. For simplicity, we will refer to the disorder as autism or ASD. The prevalence of ASD is 1 in 150 children, 3.7% in the years 2000 to 2002, but it is increased to 1 in 68 in 2010, so 1.4%. Interesting. Does it seem like a smaller number? Whatever. Okay, so rates in North America, Asia, and Europe are similar, averaging about 1% and 2%. At least some of the increase can be attributed to its improved detection, broader diagnostic criteria, and doctors' willingness to use the label because of decreased stigmatization of autism. Because the diagnosis will qualify the family for increased services and financial assistance. Some observers believe the increase is real and due, for example, the rising environmental toxins, but the position of most authorities is that we simply don't know how much the actual rate has increased or why. The good news is that there is no further increase between 2010 and 2012. Oh, okay, so got it. I'm still going back to that initial stuff. Okay. But it's too early to tell whether that signifies a trend. One statistic is not in dispute. The prevalence is almost 5 times higher among males than females. Cognitive and social impairment. In the study of cognitive abilities with children with ASD, 55% have an intellectual disability, and 16% had moderate to severe disability with IQs below 50. Impairment was not universal. However, 28% had an average intelligence, and 3% scored above 115. Autistic individuals share a common core of impairment in communication, imagination, and socialization. Trouble understanding verbal and nonverbal communication often makes testing difficult, raising questions about the meaningfulness of test results. In some cases, nonverbal tests such as the Raven Progressive Matrices are used, and the IQ is estimated from an assessment of adaptive behavior, but these do not eliminate the deficits. Difficulty with imagination is also common in the form of an ability to pretend or to use make-believe situations. Use of language is also very literal. Can you pass me the salt is met with yes, with no compliance. And in some cases, this literalism extends to the obsessive interest in facts like that scene in the movie Rain Man. These characteristics make it difficult to socialize with others, which is what sets apart children with autism the most. They usually prefer to be alone and ignore people around them. Their interaction with others is often limited to requests for things they want. They otherwise treat people as objects, sometimes even walking or climbing over them. Verbalization is usually limited, but the child often repeats what others say. Echolalia. Some researchers believe that much of the social behavior is that the person with autism lacks a theory of mind, the ability to attribute mental states to oneself and to others. In other words, the autistic person cannot infer what other people are thinking. One man with autism said that people seem to have a special sense that allows them to read other people's thoughts. An observant autistic youth said people talk to each other with their eyes. That is what they are saying. What is it that they are saying? In a study that measures this deficiency, children watched hand puppet Anne remove a marble from the basket where the puppet Sally had placed it, and put it in a box while Sally was out of the room. On Sally's return, children were asked where she would look for the marble. Normal 4-year-olds had no problem with the task, nor did Down syndrome children with a mental age of 5 or 6. But 80% of the children with autism, with an average mental age of 9, answered that Sally would look in the box. There are 2 hypotheses as to how we develop a theory of mind. According to the theory, nice, we build hypotheses over time based on our experience. Simulation theory holds that we gain insight into people's thoughts and intentions by mentally mimicking the behaviors of others. This view gets some support from studies of the mirror neurons, which we talked about in chapter 8 and 9. Individuals who score high on a measure of empathy tend to have more activity in the mirror neurons. Researchers have suggested that impaired mirror functions reduce the autistic person's ability to empathize and learn language through imitation. Not yeah, you know what I mean. Children with autism engage in less contagious yawning than others do, and they show neural deficiencies during mirroring tasks. For example, they can imitate other facial expressions, but mirror neuron activity while doing is either delayed or non-existent. Other studies showed reduced activation in the inferior frontal cortex, motor cortex, suggesting weakness in the dorsal stream connections that provide input in those areas. This interpretation was supported in the study of individuals diagnosed with Asperger's syndrome, the former term for high-functioning autism. When they imitated facial expressions, transmission over the dorsal stream, the occipital to superior, temporal to posterior, parietal to frontal, was delayed by 45 to 60 milliseconds compared with normal controls. Okay, so brain abnormalities in autism. Autism was long thought to be purely psychological in origin because no specific brain defects had been found. The problem was blamed on a lack of maternal bonding or a disastrous experience of rejection that caused the child to retreat into the world of aloneness. But evidence could be found for this kind of influence. Autistic children often had exemplary homes, and children with extremely negative experiences did not become autistic. The frequent association with intellectual disability and epilepsy implied that autism was a brain disorder. Later work found subtle but widespread brain abnormalities, especially in the brain stem, the cerebellum, and the temporal lobes. The location of damage varied among individuals, which suggested that there are various pathways to autism. There are many abnormalities in the brain of individuals with autism, but the most striking characteristic is the atypical sequencing of brain development. The autistic brain undergoes dramatic growth during the first year of life, and the overgrowth focused in the frontal and temporal areas that are important in the social, emotional, and language functioning that are impaired in the disorder. The excess growth ends around 3 to 5 years of age. But then, But by then, the brain has already reached the normal adult size. In adulthood, these areas are underactivated during tasks that would ordinarily engage them. For example, when viewing animations that require the viewer to understand the mental state of the actor, those sorts of things. Despite comparable overall brain volume, adult patients have increased cortical volume and thickness and more gray matter in the prefrontal and temporal areas, with reductions in other areas. Several studies have reported white matter connectivity reductions in the brain of individuals with ASD, particularly in the corpus callosum and need to highlight, and other long-distance connections. These findings have led to the hypothesis that underconnectivity is a fundamental characteristic of the disorder. However, additional researchers have reported hyperconnectivity. One thorough study replicated its results with 2 different methodologies and found strong evidence of overconnection in both short and long distances in the brain. An interesting observation that supports an association between ASD and hyperconnectivity. Synesthesia is found in 2.6 times more of the people with ASD than with the general population. As we saw in Chapter 10, that evidence favors hyperconnectivity between sensory areas as an explanation. So, how do we reconcile these disparate results? Studies by researchers at the San Diego State University suggest 2 possible reasons. One study reported frontal underconnectivity in milder cases of ASD to temporal occipital overconnectivity in more severe cases. Indeed, Supakur and his colleagues noticed that children with greater functional connectivity had more severe social deficits. More severely affected individuals are less likely to be included in studies because of their inability to cooperate with the scanning procedure. The other study found underconnectivity in several areas, including prefrontal cortex, but hyperconnectivity in the temporal lobe. Researchers pointed out that the underconnectivity occurred in the earlier maturing parts of the cortex, and the overconnectivity was associated with the later maturing areas. This study raises the possibility of both under and overconnectivity occurring in varying degrees within an individual, or an individual exhibiting one or the other, in both cases depending on the time progression of the disorder. Biochemical abnormalities in autism. As researchers looked for the genetic links to autism, several of the genes that turned up pointed to the serotonin, glutamate, GABA, and oxytocin. Serotonin has received considerable attention because of its contribution to neurodevelopment. Prenatal serotonin activity is regulated by cortisol levels, and cortisol is increased in the mother by psychological stress. Depression, type 2 diabetes, inflammatory disorders, and obesity. Neuroimaging studies have found diminished serotonin synthesis in children with ASD, and treatment with serotonin reuptake inhibitors, such as Prozac, decreased repetitive obsessive behaviors in autistic individuals. The antipsychotic drug Risperidone antagonizes dopamine as well as serotonin. Its strong therapeutic track record implicates that neurotransmitter. So, dopamine as well. According to the analysis of 22 previously published studies, several autism symptoms during treatment with Risperidone. High levels of the excitatory transmitter glutamate have also been found in autism. Treatment with a glutamate antagonist alone have produced unremarkable results. But pairing the anti-Alzheimer drug memantine with Risperidone decreased irritability more than Risperidone alone. So in Chapter 7, when we saw that oxytocin facilitates bonding and social regulation, for that reason it is known as the sociability molecule. Most studies have found that oxytocin is reduced in individuals with autism. Though not all of them. Even studies using a single dose of oxytocin to report improvement, have reported improvements in repetitive behaviors, self-injury, empathy, social interaction, and ability to recognize mental and emotional state from photos of the eyes. Results of an fMRI study indicate oxytocin improves trust by reducing activity in fear areas. In the amygdala and the midbrain. 2 studies with high-functioning ASD patients, oxytocin improves brain functioning by increasing previously deficit activity in the right anterior insula during an emotional interference task. And increasing functional connectivity between the anterior cingulate cortex, turning the page, cortex and the dorsomedial prefrontal cortex. Research with mice indicate that oxytocin enhances social reward by increasing serotonin activity in the nucleus accumbens. Oh, that's with mice. Okay, got it. The environment and autism. Let's see. Parental treatment has been ruled out as the cause of autism. But numerous other environmental conditions have been identified as contributing factors. One's suspicions immediately turn to the environmental pollutants, such as those generated by automobile traffic, agricultural practices, and industrial activity. Researchers generally assess such effects by studying people living near agriculture fields or a busy highway. But two studies are notable for employing more meaningful measures of pollution. Using localized estimates of traffic pollution based on Environmental Protection Agency data, traffic volume, and climate conditions during each child's gestational period and the first year of life, researchers found that living in homes where traffic pollution is highest tripled the rate of autism. Recognizing that ASD occurs in geographic clusters. Oh, that's interesting. University of Chicago researchers used country-level data on genital malformation in male children as a surrogate for environmental exposure and found that the incidence of ASD increased 283% for every percentage point increase in the incidence of malformations. Yikes. The environmental influences originate from the mother herself. For example, infection in the second trimester of pregnancy tripled the chance of bearing a child who would later be diagnosed with ASD. 10% of women who have a child with autism have immune molecules in the blood that react with proteins in the brain. This rate is 4 times higher than the general population of women childbearing age. Other researchers identified 6 brain proteins and anti-brain antibodies attached to it. All of them are involved in neuronal development. Maternal metabolic conditions, including obesity, diabetes, and hypertension, may also be associated with ASD. Mothers of autistic children were 40% more likely to be obese and 47% more likely to have any one of these conditions. Oh. Okay, the obesity, diabetes, hypertension, all those. Okay. The news is not all bad. Mothers who took folic acid during pregnancy were half as likely to bear a child who would later be diagnosed with autism. In addition, the accompanying application explains that there is no evidence that vaccines are linked to autism. So let's go there. Application, childhood vaccines and autism. In 1998, Andrew Wakefield and several colleagues published a paper in which they linked the measles, mumps, and rubella vaccine, the MMR, to autism. Years later, 2 reviews of all of the available studies concluded that there was no credible link between autism and the MMR vaccine, or a mercury-deprived preservative, thimetrazole, used in some vaccines. In early 2010, the General Medical Council, which oversees doctors in Britain, decided that Wakefield's study was methodologically flawed and that he had acted unethically. The Lancet, which published the study, called the report the most appalling catalog and litany of some of the most terrible behavior in any research, and took the rare action of withdrawing the study. More recently, a comprehensive meta-analysis examined scientific data from more than 1.25 million children that concluded that there was no relationship between vaccines and components of vaccines or multiple vaccine administrations and the development of autism and autism spectrum disorder. Many parents whose children's first signs of autism coincided with the round of the child's vaccines, which they find these reports unconvincing. More than 5,000 families have brought suit. A U.S. federal court has denied the claims in 4 cases, but that does not signal the end of litigation or the controversy. In the meantime, there is a disturbing decrease in the rate of childhood immunization in several countries in spite of the health risks, leading to large outbreaks of measles and other childhood diseases that could have been prevented. Heredity and autism spectrum disorders. Siblings of children with autism are 25% more likely to be diagnosed with autism than other children. The number would be even higher, but parents tend to stop having children after their first autism diagnosis. For the identical twin of a child with autism, the risk of autism is 60%. However, relatives frequently have autistic-like cognitive and social characteristics. These symptoms are also considered the concordance for identical twin jumps to about 92% compared with 10% for fraternal twins. In the largest study to date, researchers examined data from all 2 million children born in Sweden in 1982 and 2006, and they estimated the heritability of ASD to be about 50% and autism for about 54%, with the remainder due to non-shared or non-familial environmental influence. As you might expect, genes that have been identified contribute to neuron development and migration, synapse formation, and neurotransmitter activity. For example, several genes that code for synaptic proteins that have been implicated. Synaptic proteins are required for the formation of functioning of the synapses. Genes like genes for GABA A and GABA B receptors, and for both AMPA, and NMDA, glutamate receptors, have also been linked to ASD, and the GABA B receptor agonist improves social function and behavior. But once again, it has been difficult to identify the specific genes involved. So-called common variants, genes that occur in 5% of the population, have weak effects individually, requiring multiple hits to have an effect. Small effects is not the only reason identifying autism genes is difficult. A recent genetic analysis estimates that half of all autism cases are the result of de novo mutations. There is a limited window of opportunity to identify genes because the mutations appear spontaneously and typically are not passed on because the individual are unlikely to reproduce. Of course, oxytocin genes come into play as well. Several studies have reported a relationship between oxytocin receptor genes, the OXTR, and ASD. Evidence suggests that the downregulation of that gene and some groups in some subgroups of ASD results in increased activity in the temporal parietal junction, which participates in social perception. Also, individuals with ASD who had a mutant allele of the CD38 had lower oxytocin levels than others with ASD. Irene Voynagou and her colleagues in 2011 identified over 500 genes that were expressed at different levels between the frontal and the temporal cortices of healthy brains. Only 8 in ASD brains. ASD genes are particularly expressed in superficial layers of the cortex, where they interfere with the development of connections between the layers and between the hemispheres. As you have seen before, environmental influences almost certainly exert their effect through epigenetic modification of gene expression. This fact offers a glimmer of hope that the drugs designed to reverse these modifications might be used as a treatment for autism. In addition, important factor, I can't get it out, is understanding genetic influence in autism and other disorders, and that some genes exert control over many other genes. The RORA gene codes for the protein RORA1, which regulates the activity of more than 2,500 other genes. 2,500? Yeah, that was probably right. Some of these are involved in neuron and synapse development and synaptic transmission, and at least 6 have been associated with ASD. Researchers attempting to explain the higher incidence of ASD in males have most often looked at the X chromosome, but have little luck pinning down the responsible genes. But now researchers at Emory University have a promising candidate. In their study, mutations of the AFF2 gene occurred 5 times more frequently in boys with ASD than in boys without ASD. Male vulnerability may not be the whole story. However, females may enjoy some protection. Girls with ASD had a very long copy number variations, differences in how often a segment of DNA is repeated. That's what that means. More often than boys with ASD. This implies that it takes more mutations to produce ASD in females, which suggests a female protective model. Although cortical levels of the RORA protein are decreased in both males and females with autism, levels are higher in unaffected females than in unaffected males. The apparent reasons that the RORA gene is upregulated by estrogen and downregulated by testosterone. We're also learning that male-female differences in vulnerability may be qualitative as well as quantitative. One study found that adult males and females with autism are markedly different patterns of white and gray matter increases compared to the neurotypical control. Autistic savant and high-functioning autism. A savant is a person of exceptional intellectual skill beyond the level of typical genius, like Leonardo da Vinci or Albert Einstein. However, the term is more frequently used to describe individuals who are one or more remarkable skills whose overall functioning is below normal. Half of these individuals with islands of exceptional capabilities are autistic savants. Some can play a tune on a piano after hearing it once. Others can memorize whole books, while others can take cube roots of large numbers in their heads or calculate the day of the week for any day thousands of years in the past, present, or future. Stephen Wilshire's remarkable talent for agricultural art after brief helicopter flights had produced a detailed range of a 4 square mile or 10 square kilometer area of London and a 98-foot long or 30-meter long mural of Hong Kong's Victoria Harbour and the surrounding cityscape. A few ordinary individuals can perform similar feats, but savants' performances are typically faster, more automatic, and without insight as to how it can be done. A savant's exceptional capacity may be limited in scope. However, some who are calendar calculators cannot even add or subtract with accuracy. The source of autistic savants' enhanced ability is unknown. Dahini in 1997 suggests that it is due to intensely concentrated practice, but more typically the skill appear without either practice or instruction, as in the case of the 3-year-old autistic girl who began drawing animated and well-proportioned horses in perfect perspective. Alan Snyder and John Mitchell in 1999 believe that these capabilities are within all of us and are released when the brain centers that control executive or integrative functions are compromised. This, they say, gives the savant access to speedy lower levels of processing that are unavailable to us. By lacking the executive functions, savants perform poorly on similar tasks that require higher order processing. The idea gains some credibility from the case of a man impaired in his left temporal and frontal areas by dementia. Despite limited musical training, he began composing classical music, some of which was performed publicly. The best-known savant is Kim Peek, the model used by Dustin Hoffman in his portrayal of Raymond Babbitt in Rain Man. Peek's brain had several abnormalities, particularly in the left hemisphere. A few researchers have attempted to unleash savant-like capabilities by using transcranial electrical or magnetic stimulation to inhibit neural activity in the frontal temporal area. Most of the results have been unremarkable, but 40% of the subjects receiving excitatory right hemisphere stimulation, along with inhibitory left hemisphere stimulation, were able to solve a problem that none of the control subjects could solve. Whatever the explanation, phenomenon adds to the argument that intellectual ability involves multiple and somewhat independent modules. If the savants have an island of exceptional ability, autism is an island of impairment in high-functioning individuals with ASD. As an infant, Temple Grandin, who would stifle and attempt to claw her way out of her affectionate mother's arms. She was slow to develop language and social skills, and she would spend hours just dribbling sand through her fingers. A speech therapist unlocked her language capability, starting with slow emergence towards a normal life. Even so, she did not develop decent language skills until the age of 6 and did not engage in pretend play until she was 8. As an adult, Grandin earned a doctorate in animal science. She also teaches at Colorado State University and designs humane facilities for cattle while lecturing all over the world on her area of expertise and on autism. Still, her theory of mind is poorly developed, and she must consciously review what she has learned to decide what others would do in social situations. She says that she is baffled by relationships that are not centered on her work, and she feels like an anthropologist on Mars. I have actually read her work before. Attention Deficit Hyperactivity Disorder. ADHD is characterized by impulsiveness and inability to sustain attention, learning difficulty, and hyperactivity. Behaviorally, we see fidgeting and inability to sit still, difficulty task organizing, distractibility, forgetfulness, blurting out answers in class, and risk-taking. Diagnosticians recognize three types of ADHD. Combined with attention and hyperactivity, impulsiveness is most common. But individuals are predominantly inattentive and predominantly hyperactive impulse. ADHD is the most common among childhood-onset behavioral disorders. A meta-analysis of 179 studies estimated prevalence of 7.2 worldwide. Boys are more than twice as likely as girls to be diagnosed. Although ADHD is often thought of as a learning disorder, and many children with ADHD do have at least one learning disability, the effects are often felt in every aspect of a person's life. Besides having unexpected difficulties in life and work, these individuals have greatly increased rates of antisocial personality disorder, criminal behavior, and drug abuse. In fact, 35% of people seeking treatment for cocaine abuse have had a childhood history of ADHD. People with ADHD are twice as likely to die prematurely from accidents. Drugs are usually prescribed to control the symptoms of ADHD, and from 2002 to 2010, the prescription rate has jumped 46% in the United States. Dang. While in Denmark, it went up a whopping 630%. Denmark. With 69% of diagnosed children taking medication, some observers have raised concern that children are being overdiagnosed and over-medicated as an easy solution to classroom behavioral problems. Agreed. The link with drug abuse fostered concern that treating children with stimulant drugs such as methylphenidate or Ritalin or amphetamine was leading to addiction later on in life. However, research followed children with ADHD into adolescence and adulthood and revealed that at worst, stimulant treatment made no difference, and that it might even be protective against drug abuse. The more positive outcomes for the treated individuals could have been due to the reduction of symptoms, or it could have been the result of other factors, such as the support of parents who opted for treatment, but at least these results do not support fears that medications act as a gateway drug. Although ADHD is considered a childhood disorder, in a 13-year-long study of 2,040 twins in the UK, 22% of the children diagnosed with ADHD continued to meet the criteria by the age of 18. The DSM-5 diagnostic criteria require onset during childhood. But 68% of the 18-year-olds who met the diagnostic criteria without regard to age of onset had not done so in any of the 4 previous screenings. These results were not unique to the United Kingdom. They were confirmed in a large study in Brazil and are supported by earlier study in New Zealand. Unfortunately, the data cannot tell us whether adult-onset ADHD is a different form or if these individuals were simply able to compensate for their impairments throughout childhood. We do know, however, that the adult-onset ADHD group had higher IQs than the childhood-onset group and was preponderantly female. Interesting. All right. Brain abnormalities and ADHD. Numerous studies have reported one difference or another in the brains of individuals with ADHD, but the results have been inconsistent. Brain MRIs cost anywhere between $1,000 to $5,000, so study samples are usually small. Brain differences in psychological disorders are typically slight, and the order of just a few cubic centimeters of volume. So reliable detection of these differences requires large samples. Now, a 9-country international group has solved that problem by compiling data from 1,700 people diagnosed with ADHD and 1,500 people without. They found reductions in overall brain volume and in 5 specific structures involved in emotion, motivation, and reward processing: the caudate nucleus, the putamen nucleus accumbens, the putamen, I don't know if I'm saying that one right, nucleus accumbens, amygdala, and hippocampus. The amygdala had the most pronounced deficit, which is important because the amygdala functions not only in emotion, but also in response inhibition. Reductions were greatest in children and leveled off in adults, which lent support to the brain maturation theory of ADHD. White matter integrity is also reduced in children with ADHD in both interhemispheric connections and in anterior-posterior pathways. They share this characteristic with children diagnosed with ADHD and obsessive-compulsive disorder. In fact, the children with a greater white matter impairment also had more impairment in functioning regardless of their diagnosis. Because of this reduced connectivity, activity is segregated into localized clusters rather than integrated across larger areas. There is a hub of local connections centered around the amygdala, and research suggests that the ADHD brain may be configured to respond impulsively instead of having neuronal organization to regulate and inhibit responses. Other studies have found overall reductions in brain volume, particularly in the prefrontal and parietal temporal areas, the cerebellum, along with reductions in cortical thickness. The reductions were observable in adults at 33-year follow-up, though there was some evidence of mitigation of the deficiencies in those that were symptom-free. Genes, neurotransmitters, and ADHD. To say that ADHD runs in families would be an understatement. Heritability averages 76% across studies, and the concordance has been variously estimated at 58% and 83% for identical twins, and 31% to 47% in fraternal twins. Like autism, ADHD is a complex, multi-symptom disorder, and different individuals display different combinations of symptoms. Whole genome-wide studies have implicated genes involved in cell migration, synaptic excitability, and neuronal plasticity. Another study of 222 copy number variations, or CNVs, in ADHD patients were not found in control subjects. These CNVs were in and near genes involved in synaptic transmission, neural development and learning, and other psychological functions. Numerous CNVs are in chromosomal locations that have been identified in autism and schizophrenia. Participation of a gene in more than one disorder should not be surprising, since different disorders share some symptoms. And remember, we are not talking about a gene for autism or a gene for ADHD or a gene for schizophrenia, but genes that regulate brain growth, receptor development, and so on. However, the genes that are most frequently implicated in ADHD are involved in neurotransmission. In fact, it is difficult to discuss the genetics of ADHD without simultaneously discussing neurotransmitters. A review of genetic studies identified 24 hot genes, which have been linked to ADHD in at least 5 studies. The large majority of these genes are involved with dopamine, serotonin, and norepinephrine transmission, including the synthesis, transport, and receptors. In addition, one gene's protein contributes to the construction of the nicotinic acetylcholine receptor, for the past half century. ADHD has been treated mostly with stimulant drugs like methylphenidate and amphetamines. These drugs increase dopamine and norepinephrine activity by blocking the reuptake at the synapse, which supports the role of both transmitters. Most research, however, links ADHD to reduced activity in dopamine pathways, including the prefrontal cortex and the striatum. These structures' functions include executive control, impulse, inhibition, working memory, movement, learning, and reward, all functions that are affected in ADHD. The significance of the reward may be less immediately obvious, but several researchers believe that impaired reward contributes to the impulsiveness because the allure of later rewards is too weak to overcome the temptation of immediate gratification. Some 20 to 30% of patients do not respond to the traditional medications or cannot tolerate them. Two drugs sometimes used in their place, modafinil and atomoxetine, block norepinephrine reuptake, adding support for its role in ADHD and as suggested by genetic studies. And also modafinil is the non-narcotic helper friend from my personal experience. The environment in ADHD. What appears to be an environmental cause of ADHD often turns out to be an indication of a genetic predisposition in parents. This includes both correlations of ADHD and with maternal smoking and stress during pregnancy, parental abuse of alcohol, stimulants, and cocaine, and parental mood and anxiety disorders. Confirmed environmental influences include brain injury, stroke, complications during pregnancy and birth, along with some toxins. Lead, for example, is a known neurotoxin, although eliminating lead in gasoline and paint has reduced levels in the environment. It occasionally is found in children's costume jewelry and imported candies, as well as in the soil and water. Okay, so it's just like everywhere. Okay. A recent study confirmed higher blood levels in lead in children diagnosed with ADHD, and lead levels were corrected with teacher-parent ratings of symptoms. Another source of concern is air pollution. Children whose mothers were exposed during pregnancy to high levels of polycyclic aromatic hydrocarbon from sources such as traffic and fossil fuel burning, fossil fuel burning electric plants have increased levels in ADHD symptoms, especially inattentiveness at the age of 9. As with autism, another culprit is the organophosphate pesticides. Children with above average urinary levels of the metabolite of these pesticides are twice as likely to be diagnosed with ADHD compared with children with undetectable levels. Even chemicals found in cosmetics, perfumes, and soaps are turning out to be a problem. Mothers who had higher levels of phthalates in their urine during the third trimester of pregnancy reported that their children had significantly more trouble with attention, aggression, and depression. How phthalates cause, I think I'm saying that right, phthalates, cause this result is unknown, but they may disrupt thyroid hormones, and that is important during brain development. Although these examples are distinctly environmental, it is important to remember that environmental insults often exert their influence by changing gene function. For example, inducing concussions in rats altered over 1,200 genes. 16 of them are linked to ADHD. Another example is dietary. High fat and sugar in women's diets during pregnancy was associated with increased methylation of the IGF-2 gene, which is involved in the development of brain areas implicated in ADHD, along with higher symptoms of ADHD. In addition, genes can influence whether an environmental condition has an effect. In children with ADHD who had a specific mutation in the C28.2Y gene, safe levels of lead exposure were associated with increased symptoms. So even the safe levels with those who had the mutation on that gene. All right, so in perspective, it is important, as important as the assumption of some assessment of intelligence is in our society for determining placement in schools, opportunities, and for continued education, and employability and promotability. It is remarkable that so much disagreement remain about what intelligence is. This lack of agreement makes it more difficult to study the brain functions that make up intelligence. Nevertheless, we have identified several features that appear to contribute to a greater mental power, brain size, neuronal conduction speed, processing efficiency, and working memory are among these. Although it would be an error to over-localize any function of the brain, we also know that some areas have a special role in important cognitive functions related to intelligence. It remains to be seen that whether any characteristic of these areas, such as size,
explains why some people have a particularly strong talent in one area, such as creative writing or mathematics. Some people hope that we will eventually have objective brain measures that will tell us exactly how intelligent a person is or whether the child has ASD. When we reach that point, perhaps another dream will be realized, the ability to diminish or even reverse some of the effects that rob the intellectual disability, the autistic, and the aged of their capabilities. We may even be able to increase the intelligence of normal individuals. We can only hope that our capacity to make the ethical decisions required keeps pace with our ability to manipulate the human condition.